Hello and welcome back friends, and welcome to BioniNirmal. And in this video tutorial we will be talking about the transcription, the basic concept. Before going to discuss about the total phases of the transcription, first of all we should have the clear concept about the gene and also the few properties of the DNA and also the process of the transcription, so that I am in this video tutorial I am going to give you the basic concept about the transcription, and in the next video I shall elaborately describe you about the process of transcription. Actually the transcription is actually the process by which all kinds of the RNA are actually synthesized. mRNA, rRNA and the tRNA, these three types of the RNA are actually synthesized by the process of transcription. And in cases of the eukaryotes, there are presence of the nucleus, and inside the nucleus the transcription process is actually occurred. Okay, so if this is the nucleus, so this is a eukaryotic cell and this is the nucleus, and in the nucleus there are presence DNA. Actually DNA cannot move from the nucleus, and the DNA reside inside the nucleus, and so that from this DNA the information from the DNA actually transmitted into the mRNA. Okay, and this mRNA actually come out from the nucleus by a protein which is called X protein. So there is a protein which is called as the X protein, and this actually and through this protein mRNA actually come towards the cytoplasm. That is, this actually come out from the nucleus, and inside the cytoplasm this mRNA actually combined with this ribosome, and by the process of the translation this will produces the protein. Okay, and in this video tutorial I shall tell you that how this mRNA is actually synthesized from the DNA. Actually the information of the DNA actually enters into the mRNA, and this mRNA actually carry the messages, a message from the DNA, and inside the ribosome by the process of the translation this protein is actually synthesized. So that the synthesis of the mRNA is actually very important, because the protein synthesis is actually occurred in the ribosome, and which is reside in the cytoplasm, and this mRNA actually come out from the nucleus into the cytoplasm. Okay, and so that by the process of the transcription, actually the only where the in cases of the replication the many enzymes actually was acting, but in cases of the transcription only the single enzyme, that is the RNA polymerase, actually control the overall process. Okay, a single enzyme. So a single enzyme in cases of the prokaryotes. So in prokaryotes, single enzyme, and this enzyme is called RNA polymerase. So this RNA polymerase is actually responsible for synthesis of all kinds of the RNA, and this RNA polymerase is called the holoenzyme, and this holoenzyme is consisting of core enzyme and sigma factor. So this holoenzyme is actually consisting of core enzyme plus sigma factor. Okay, so sigma factor, and this core enzyme is made up of two alpha subunit, one beta and one beta dash subunit, and sometimes this is made up of omega subunit. Okay, so if this is the alpha subunit, so these are alpha subunit, and this is beta and beta dash subunit, and inside this there are presence of the omega subunit, and the sigma factor is actually present here. Clear? Actually the sigma factor is actually recognize the promoter. So sigma factor recognize the promoter in cases of prokaryote promoter, and this actually continue, this core enzyme actually continue the process. So this is the function of the RNA polymerase in cases of the prokaryotes, and later I shall describe you about the total function of the RNA polymerase. But in cases of the eukaryotes, the three RNA polymerases are required, and these three RNA polymerases are RNA polymerase 1, RNA polymerase 2 and RNA polymerase 3. but eukaryotic RNA polymerase. So eukaryotes, RNA polymerase 1, RNA poly 1, 2 and 3 are responsible for transcription, and by RNA polymerase 1 the rRNA is synthesized. RNA polymerase 1 is responsible for synthesis of rRNA except 5S rRNA. So 5S rRNA. So RNA polymerase 1 is responsible for synthesis of the 5.8S rRNA, 18S rRNA, 28S rRNA. So these all types of the RNA are actually synthesized from RNA polymerase 1. RNA polymerase 2 is responsible for synthesis of mRNA, and RNA polymerase 3 is tRNA, and 5 is rRNA. So these are the function of the RNA polymerase, and so these are the functions of RNA polymerase I have described you. In cases of the prokaryotes, single RNA polymerase is responsible for transcription, but in cases of the eukaryotes, 3 RNA polymerases are responsible for transcription. And BioniNirmalghosh I actually told that one gene and one enzyme hypothesis, that is the gene is responsible for synthesis and enzyme, and this is actually called as one gene one enzyme hypothesis, and this hypothesis was proposed by BioniNirmalghosh I, but later Ingram actually told that one gene is responsible for synthesis of one polypeptide chain. Anyway, and this is the most modern concept. So according to Ingram, one gene, one polypeptide concept, one gene, one polypeptide. So one gene is responsible for synthesis of one polypeptide. Clear? So gene is a portion of the DNA and which is responsible for synthesis of the one polypeptide chain. The starting point of a particular gene is called the promoter. So this is the promoter or start of a gene, or start of a gene, and terminator, and this is the ending of a gene. So terminator is the end of a gene, and in between the promoter and the terminator there are presence of coding region, so coding portion of the gene, and this portion is actually transcribed. Okay, and this portion is easily transcribed, and later I shall describe you about the promoter and the various sequences of the promoter, and which kind of the sequences present in the terminator. Okay, but here I can tell you that the DNA DNA is actually have the two strands and which are anti-parallel, that is 5' and the 3' strand, and since the RNA is single-stranded, so only one strand will be copied. So only not copied. So this is transcribed actually. So only one strand will be transcribed. And now let us examine that which strand will be transcribed. I am going to tell you that which strand, that is 5' to 3' strand, or 3' or 5' strand, and which strand will be transcribed actually. So the DNA actually has two strands. So if this is the DNA and here is 5' strand and this is 3' strand, and there is 3' strand and 5' strand, actually transcription proceed in 5' to 3' direction. Okay, so that 3' to 5' strand will be transcribed. So this strand will be transcribed. So the mRNA, if this is mRNA, this is 5' to 3', and this is mRNA, and the strand which actually serves as the template, and this is called as the template strand, and this strand is called as template. Okay, and This strand is called template, and which strand will be transcribed, or this is called antisense strand, or this is called antisense strand, or this strand is called non-coding strand. Okay, so this strand is called non-coding strand, and the another strand, and this strand, that is the 5' strand, and this is called as the non-template strand, or this is called same strand, or this is called coding strand. Okay, so coding strand has same sequences of mRNA, except instead of thymine there are presence of uracil, except instead of thymine there are present uracil in mRNA. Okay, and if this sequence is suppose this is A, T, G, C, G, 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 etc., and so that the mRNA will be in cases of this is the complementary bases will be thymine, but in the mRNA the thymine is not present, so this is the uracil, and A, this is C, so this is G, this is C, C, etc., and here this will be thymine, and this is adenine, this is guanine, and this is also guanine, and this is also this is cytosine and cytosine. So this coding strand has the same sequences of that of the mRNA. You can see that instead of the there are presence of the uracil instead of the thymine. So if in the DNA there is presence thymine, and here is the same strand of the mRNA. So this is actually, this will be cytosine and this will be guanine, and so this is actually the story about the template and the non-template strand. And so this is the basic concept about the transcription, and in the next video I shall describe you the details process of the transcription. So that's it guys, and if you like my videos then you can share my videos and you can subscribe my channel BioniNirmal for more updates.